Hello guys, welcome back to Miyaba Academy, and today I wanted to teach. So here's me trying to teach you guys something that I recently learned and I have applied in various fields. I have used this particular website to calculate scores and to determine the pathogenicity of my variants that I was given for an actual person, not just for research but for um, a person with a disease. He got his whole genome sequencing results and he wanted me to analyze them, and this is one website that I used to record the scores. And yeah, it has been a while since I last posted a video, and that's because I had a lot on my plate. I had papers to write, I had classes, and I just wasn't feeling it. Like, I wasn't really into teaching at that moment. Now, I think I have learned enough to go around and teach people myself. Yeah, and this is actually interesting. Like, I love my degree. I'm currently doing my bachelor's in genetics. This is the fourth year of my bachelor's, and so far we've learned a lot. I feel like we've learned more of molecular biology than of genetics. But yeah, so I think this comes in bioinformatics or genetics, calculating scores for variants. So why do we calculate scores to begin with? So basically, what these scores do is that they look at a mutation, a genetic mutation, and they, for example, you get a mutation in a gene, a nucleotide from switches from C to A. So what it does is that it changes the sequence, which in turn changes the sequence of the protein. Hence, we get an effect. This effect can be pathogenic. It's classified as pathogenic according to ACMG. So yeah, I'll talk more about variants later because the main goal of this video is to calculate scores. Yeah, I don't want to prolong this. So yeah, beginning with an example that I picked up. So this is the name of the gene that was affected in a patient. This is the KCNQ3 gene, and I tried to add as little information as I could so that I could help you how to get more information when you just provided with a SNP. So basically, this is the SNP that has been provided to us. We're gonna copy it and head over to Ensembl's VEP. So what is VEP? Basically, VEP stands for Variant Effect Predictor. This is the website that calculates the effect of changes and mutations in a gene. So there are various ways you can paste the information. We have the RSID. That's how we're gonna do it. We could do it with other ways. We could do it with the chromosome number, genomic coordinates, nucleotide substitutions, and we could do it that way. Chromosome position, chromosome number, position, replacements. This is how we're gonna do it today with the variant identifiers. We call it RSIDs. This is also how you do it. This is the transcript number and the change in the nucleotide. So if you want to do that way, I'll tell you in a bit. And lastly, again, chromosome position and the change in nucleotides. So this is our RSID. So if you want to get more information on the RSID, yeah, I also had to tell you that. So you go to the Ensembl website. I love this website personally. I mean, we could use other websites like UCSF genome browser, but I prefer using this website because it is very compact. I just feel like it's not everywhere. We could use other websites, but I preferred this website at the moment. So this is the genome it got it from, the variant ID. It's a SNP. A SNP is a single nucleotide polymorphism. So this is the reference allele and that is the alt allele, meaning this is the healthy person's allele and this is the allele that changes when you get the mutation. This is the frequency, the CAD scores. Here is the chromosome number and then the genomic coordinate. I think that's all this has to offer. Yeah, so here is the ENST number and the ENSP number. So yeah, moving over to the website. I'm sorry I'm stalling this, but I feel like I have so much to tell you guys. So you could throw in a file or a URL, but I prefer pasting my SNPs when I have less data. So you go here and you click wherever you want. I want the protein, UniProt ID, HGVS. We can get our HGVS ID. And what else would we require? It gives the main transcript as well. Okay, so protein annotation. I would need to know the protein interactions and the changes in the structure of the protein when we get the mutation, and clinical significance with the exact match. So here is the main thing that we're here for. So there's SIFT scores, so we need the prediction and the score, and polyphen, which sees the change in the protein when we get a mutation. We'll enable this. Sure, CAD scores should be enabled, and Revel. So these are the different softwares that will give you the result. Basically, there used to be a different website to calculate the scores for SIFT, but they moved over to VEP, and now it's a compact. It gives many scores in one website. I would love to see the conservation status. And lastly, we'll run it. This is the things that I've done before. So this is our SNP right now. It's in queue. It's still running. You can comment down below for other ideas. If you want to learn something, do let me know. And yeah, you get your results right here. Okay, so as you can see that this is a missense variant. It tells you that it's most likely a missense variant. So we get multiple results, right? So all of them having the same SNP, but the difference lies here. So yeah, if you want to see, I would love to see this, my protein, change in protein and change in codons, reference allele, clinical significance. Yep, SIFT and polyphen scores, these are really important at the moment. And CAD scores, yes. So yeah, as you can see, the mutation is, the consensus is that it is unsignificant. The significance is uncertain because there is no allele frequency yet. That means that not many people have this mutation. But yeah, the CIF scores do show that it is benign, meaning that this is not a pathogenic variant, that it doesn't have a detrimental effect on our genes, on the patient who has this variant. He or she, they're most likely to be not affected by this variant. So yeah, do let me know if you have any other questions. You can give me feedback. You can let me know if I'm boring. I don't know. I'd appreciate any response. So yeah, thank you.